Still.
Okay, so I think everybody can hear us now. I was saying that the slides are in the form of a handout. So we will go through a lot of them quickly. Feel free to send me an email with the title slides. You can say more things, you can say hi if you like, but slides is sufficient, and I will send you a PDF of the slides with PubMed ID references, and you can study them on your own time. As I will have already said, we will go through some material quickly. I don't have any conflicts of interest. Yes, but I do have some biases, and the biases emanate from my workplaces. I do work as a regular pediatric dentist. Mondays, half Tuesdays, and every other Wednesday, I'm either in the dental school clinics, in the sedation clinic where we see young children under pharmacological influence, or in the children's hospital where I was yesterday all day, and we treat children under general anesthesia for restoration of dental caries or even trauma. So I see that part, the end point of dental disease. But I also work, I have an appointment at the School of Public Health, and also do a lot of dental public health work, which means that we may be 2 hours away in a public preschool doing a toothbrushing campaign about showing children how to brush their teeth, and working with parents and school teachers in topics promoting about children's oral health. So I do see these two extremes, the upstream, the downstream, and also I do some research. So with a good network of collaborators, including those, and Andrea is a key collaborator. I think more than 10 years we've been working since the times of when pyrosequencing was cool. I remember that time. It's a large team of collaborators we have, supported by NIH and other funding institutes, that allows us to investigate a little bit more the fundamentals of oral health and disease. So a big thank you to the team beforehand. Today, I will focus on insights from microbiome studies, as the title of the talk says. But to get there, I would like to introduce you to the way we think in dentistry, because I know some of these things will be relatively new to you, even what dental caries is for some of you in the most fundamental level. So I'll begin initially by explaining the overall scientific and healthcare framework we talk about, that we frame our work within, which is precision health. Then I will take one step closer, and I will talk about dental caries and how do we operationalize that clinically. And then we'll talk about the microbiome studies that we've done and we're still doing in the context of precision oral health and early childhood caries or oral health. So to begin from the precision part, the precision health, many of you are aware of this general trend regarding precision medicine, precision health. The All of Us study is one big investment that the NIH has done, basically trying to realize the participation of about 1 million US of Americans who are not necessarily patients, but are participants in a mega study where they will contribute clinical information, other behavioral information, lifestyle, genetic information, saliva samples, microbiome samples, stool samples, all those. The point and the beauty of trying to move to people versus patients is that in many fields of study, including, and I can say that dental studies is one of these, unless you focus on studying populations and you look only at patients, you may be missing a big part of the picture. Because really not every person will be a dental patient. Ideally, everybody will go to the dentist every 6 months or a year for cleans and checkups. That is not happening, and especially in younger ages. So if you only look at patients, you may be missing most people, and arguably the ones that may be most informative, because otherwise you may be seeing the ones who are extreme cases, they have cavities at age 2 or 3, and we see them, or the ones that have extremely health aware and able parents to bring them to a dental appointment at age 2 and 3 for prevention. And everybody else is unobservable in our studies. So moving to studying people is a huge advantage if you really try to understand biology, lifestyle, and other big forces underlying health and disease. The other benefit of doing this precision health approach is that you may be identifying subgroups of people that respond differently to treatment, or different treatments being optimal for different groups of people. conversely. So the idea is that you're not studying the average patient or the average treatment. Arguably, the average patient doesn't exist, right? It's just a summary of everyone, but that person may not exist. It's like half male, half female, the median age, the median ethnicity or rank, that doesn't exist. So already the All of Us has made available data that you can go and access. You can go to data browser in this address and start navigating the data. Of course, if you log on to the workbench, which is the panel where you can access the data, you will only be seeing this descriptive information. So you can do actually analysis if you log on to the workbench. You have to get some permissions, but it's publicly available essentially. The problem for us is that oral health is not formally included in this endeavor. Of course, there's information coming from electronic health records like Epic and others, but dental and oral findings are not routinely included. So you'll see most frequently diagnosis about common chronic diseases, right, that you get an ICD-10 diagnosis. So unfortunately, oral health is underrepresented. The other problem is that so far the All of Us study is not including children because there are some ethical issues that they're working through, and I think they will be included. But for now, you don't have children and you don't have dentistry. So if you're a pediatric dentist, you're doubly excluded. That's a problem for us. But I'll show you later how we address it. Another interesting component of this precision medicine trend is what is really making clear what hurdles we need to overcome. This is a visual summary of a workshop we had in Bethesda. I was part of the oral health protocol development team, the All of Us study, and this was a working group of or a meeting of all working groups of all healthcare topics, let's say. And there were 4 areas that I think it was worth highlighting your attention. That summarized, many of these presentations, and this was actually one keynote presentation. The first one that we really have to agree on in the oral health community, and I can focus it perhaps in the dental research community, is that it's not that everything that we know is wrong, but so we shouldn't throw away everything. But we must acknowledge that it's incomplete. So that's the way to kind of keep advancing what we know. It's hard sometimes for people who've devoted decades of their research career into a topic to say, well, you're irrelevant now, because now we can do whole genome sequencing analysis. So therefore your candidate gene work is not relevant anymore. But no, we can say, look, what you've done is relevant. We're just going to complete it by studying the rest of the 3 billion base pairs of the genome. So that's kind of a little bit diplomatic, let's say. The other part is that we should remember for whom we work. And if you're in the NIH-funded realm, you work for the people, you work for the taxpayers. So you have to remember that what you do ultimately should provide value back to the community. So that's important to know. So it's not only academic what we're doing. It should be practical, not immediately evident, but keep that in mind. For that reason, we should also be aggressively sharing resources and know-how. I lived through an era where there was really competition for what group will publish first, and that still is happening. But even within the last 20 years, I have seen the change where now huge collaborative efforts are the way to go. People recognize faster, but when you join forces, you can really advance the field faster than working in 3 different independent groups: North America, South America, Europe, Asia, Australia, all these things. So you see more consortia forming faster these days, and they make huge advances. And the final point on the left is that concomitant with all these changes, we must allow for this disruption to happen in the medical domain, in the diagnostic boundaries, the diagnostic classification. We're learning more and more about the biological basis of health and disease. So it has to happen that the clinical diagnosis, the labels we put on things, have to be able to change. You have strokes, right? So you have hemorrhagic strokes, you have ischemic strokes. These are clinical labels. There are other biological subtypes of disease that we must allow for them to enter these taxonomies. There is resistance, and there will be resistance in the traditional medical community, but here we have to allow for that disruption to happen slowly. So what do we mean with precision health? I will go over the simple example I use when I talk about precision health with our students. And this comes from the mental health domain. So here, consider a group of people that have these overlapping symptoms-based categories of mental or psychological disorders. You can imagine frequent headaches, low energy, mild depression, other symptoms. And this is what they present to their physician with. The premise is that if we could know really the biology underlying each person in each condition, whether it is by behavioral, social, situational, you name it, genetically driven, we should be able to use that information to reclassify people in more homogeneous categories. And therefore, there's more clarity into what condition, what each condition is, what each group is suffering from. And the other premise here is that we should be able, theoretically, to approach and treat each group differently. Because one group may need counseling, another group may need pharmacological therapy, and different groups may need different medications. And the fourth group may be suffering from social issues, or there's a war in the area. There are other social things going on. So you cannot treat these groups the same. But to get to that point, you must be able to understand and measure and operationalize the other things in the middle. And the other things are not only the biology, the microbiome, for example, but you have lifestyle behaviors and social things. But this is the premise of precision health. Basically trying to get to more homogeneous, biologically refined categories of health and disease outcomes. Of course, if you cannot do the last step, which is if you cannot optimize treatment, you may say, well, it's theoretical, right? If I'm treating everybody the same, if everybody's getting one thing, why bother doing it? So of course, if you do that, you have to demonstrate that there are available improvements in care. So, but that generally follows the development of that. So I mentioned that in the All of Us study, there are no children, there is no dentistry. So in North Carolina, starting 2014, we were able to do a small precision oral health study we enrolled a little bit more than 8,000 children, ages 3 to 5, and I will talk more about it. But our general philosophy was that we would enable both population-based studies, and that is what is hidden under the Zoom banner there, and also deep phenotyping studies at the same time. So genomics, microbiome, transcriptome, metabolome, and all of those. And the idea was that this way we would not only help, say, public health narratives to get strength from biological studies, you can actually demonstrate that social adversity in some ways may be manifested in biological ways, and vice versa. Because you're doing basic science studies in the community, you have representation of everyone, and you can say that, look, this represents to some degree the population that live in this area. It's not that whoever could come to our high-level clinic, which is a radius of 20 miles. Now, how relevant is this to oral health? I will briefly mention that in the oral health domain, our thesis from the oral health side is that the first thing we must do for precision oral health, we must agree on measuring things correctly. And of course, I don't want to give you a lecture on the nuances of what dental caries is and how it's defined, but I have to say that we have to come up with valid, reproducible, and generalizable measures of dental endpoints. The second point is that we need to use multi-omics, and multi-omics here essentially is another keyword for biology, understand really the biology of health and disease, and to some degree be able to measure it in a practical way. That's where the biosensor business comes in. Of course, we may be doing these studies with thousands of teachers in the multi-omics domain, but in the biosensor business, you may end up measuring 10 things. Because you've done all the studies, you know what you need to measure, you measure 10 things using an intraoral sensor that pairs with your wearable device and gives you where you are today in the green, orange, or red, and then maybe you get it in your wearable, or your dentist gets it and then sends you a text: brush more, floss more, or eat less sugars. And that is not very far from being real life, I think. Next, we must be able to leverage big data, and big data relevant not only to population-wide studies, talked about 1 million people, But the closer you get to the biology, more you go to the omics side, the data are still big. Actually, they're bigger. They're billions of reads in the omics data we get. Social context also extremely influential. And finally, invest in science, technology, and education, and train people, mainly providers, to think in this new precision health manner. Now, now we're zooming into the dental side of things, but here I want to talk about the two most common oral diseases, so you can see where the microbiome comes into place. The two most common oral diseases are gum disease, which in that case is periodontitis, so it's a severe form of gum disease where you start losing bone support, and dental caries, cavities. Okay. So the parallels or the commonalities of the two diseases are they're both person level diseases. So for many years, people thought that cavities is what your tooth gets. You don't get it as a, so the disease was somehow assigned to the tooth. But this doesn't really make sense. In any other medical specialty, yes, you get effects on different organs or tissues or body sides, but you get as a person the diagnosis. You have alopecia, it's not that your head is getting bald, like you get male pattern baldness, like I get a diagnosis. Or have a headache. Sure, my head hurts, but I have the headache, right? or anything else. So we unfortunately have to emphasize to the dentist that dental caries is a person level disease that manifests on different teeth and tooth surfaces, but it's really a person level disease. There's an ICD-10 diagnosis code. And as a consequence of this disease, a cavity, a caries lesion process that develops. And this is important for the microbiome work that we will talk about later. The second part is that the processes that lead to these conditions are not easily observable. You don't see them. For my periodontist friends, those who treat with gum disease, it's a bit easier because they see gum bleeding. So if your gums are bleeding, you have an early form of gum disease. In dental caries, you don't see that. And you don't see that because what is happening, you're losing mineral from the tooth. So if you could see your teeth bleed somehow, what you would be seeing, the equivalent is that there's mineral escaping the tooth. Why? Because you've eaten something sweet and the microbia on your tooth surface are fermenting it to lactic acid. And that lactic acid basically demineralizes your tooth and you see that escaping. But because we don't see that, we cannot observe the process in dental caries. But if you could visualize your teeth bleed mineral, it would be easier to reverse the process because you don't wait for the cavity to develop. You see the process. Finally, they're both heterogeneous diseases. They develop in different ways between different teeth, different surfaces, populations, all this. But really, both of them, if you think about it, are defined by an interplay between a tooth and the biofilm. In gum disease, most of this is below the gum line. You still have a biofilm and a tooth and a gum surface. For dental caries, same process, just above the gum line. Tooth, biofilm, and interaction. Both of them are happening really there. And they are modified by everything else. But where the rubber meets the road is that tooth, biofilm. So they have this common. This is unfortunately a young child that has what we call early childhood caries. These are cavities in baby teeth, and that child is about 3 years old. You will wonder how can it get that bad very quickly? It does. These baby teeth are very immature. They're less mineralized than adult teeth. And once the cavity passes through the enamel, which is the hardest part, and the enamel is very thin in baby teeth, it goes to the softer part of the tooth, the dentin, very quickly. And it gets into the nerve of the tooth, the pulp, and it can create an infection. And of course, you can see a pattern where the upper teeth are more affected than the others, and that may have to do something with that child going to bed with a bottle that contains anything else than water, because that will stay in their mouth overnight, usually, or for a prolonged period of time. And also on the pits and fissures, the chewing surfaces of the molars, the back teeth. And you will also appreciate that the first molars, the ones closer to the front, are more affected than the back teeth, simply because they came earlier. So here, but again, every tooth is affected. So this condition is kind of catastrophic. we will have to extract the front teeth, the upper front teeth, and there's no replacement you can do for this child because this child is growing. You cannot put the denture, and they would not wear it anyway at age 3. Right? So you have to accept that deficit, and then you have to do extractions or other restorative work with crowns in the back teeth. So a lot of work that theoretically could be preventable. We don't think we can prevent all dental caries, but the catastrophic side of things, we think with proper education and oral health promotion, we should be able to prevent to some degree. But to characterize this condition completely, what do you really need? So it's a very complex phenomenon. You may say, well, of course, it's microbial, right? Because it's the biofilm-tooth interface that caused that, but why so much? So you may say it's really a social thing or an education thing because that family should know what to do. But then you may say, oh, you know what? Yeah, it's a single parent with 6 children and 2 jobs, and there's just no way to do it. So it gets so complex that you need to know everything, right? So in some cases, it is really a social condition, non-preventable or not immediately preventable. But still, I will say that if you wanted to define the disease, you still have to be somewhere between the tooth and the biofilm. So in that case, you have probably a very aggressively demineralizing biofilm, something that has a very acidic and aciduric and acidogenic profile that dissolves teeth in a very high rate. And when that is established, what tends to happen is that biofilm, that environment, selects for species and taxa that thrive in an acid environment, and all the good bacteria that like to stay there and protect the environment die or go away or relatively reduce in proportion. So it's very hard to control at that point. For a long time, and when I went to dental school, I was taught that dental caries is infectious. And this was driven by the observation that there are some bacteria that are found in high prevalence in dental caries cases. We were told that strep mutans was the causative agent, and maybe lactobacilli were also involved. And at some point, when there were mentions of if you're a mom, don't kiss your baby. If you have a cavity, you're going to infect them with strep mutans, and we're going to be doomed forever. We're going to destroy their life because they're going to have cavities uncontrollably forever, so don't kiss them. And I was thinking about it. I said, that's not natural. That cannot be true. So, but that was based on really incomplete, I'm going to be very respectful, the first line, very incomplete data, because back in the 90s and 80s and 70s, true, people who studied this, they could only see in a petri dish strep mutans, because strep mutans grew easily, they could isolate them, and they associated them at high proportions with catastrophic diseases. But now we know it's not one species. Actually, strep mutans is not in the top 40 or 50 anymore. There are many more species that are involved. So it's true that strep mutans is going to be there, but it's not the one causing cavities. Now we understand that really dental caries is an imbalance, it's a symbiosis. It's that shift in pathological species or unfavorable species that will be established because the environment has changed. And what's the environment is sugar, abundant sugar, lack of oral hygiene, lack of fluoride that would normally control that balance. And now the most recent understanding is that of a non-communicable disease. Took us, I don't know how many, 40, 50 years to kind of formally put it in paper, but now we understand it's really an imbalance. And there are many ways to articulate that, but it's really all about that mineral balance between, so it's a balance of demineralization, remineralization between the tooth surface and the biofilm. I mentioned earlier that now we have a better understanding of how many species are in the oral cavity, especially the supragingival microbiome. Now, if you go to the strain level, of course, you have thousands of those. But the main conceptual advancement is that now we understand it's not really a species that we're trying to find. We're trying to define the keystone pathogen for dental caries. It's not like one thing. We understand that's a community shift. And that community really selects very smartly for their environment, like in every other body site. So if you have abundance of sugars, fermentable carbohydrates, you're going to have this selection for species that thrive in an acidic environment. And they will, of course, sustain that environment, and more species that thrive in acidic environment will establish themselves. And that ends up modifying again the environment, reinforcing the environment. So this is really the shifts that we're trying to understand. It's not so much the one species that's going to cause disease. In other body sites, there are situations where you have one main species that will really shift the ecology that much and the interaction with other species that will create disease. In the oral cavity for the common forms of caries, it doesn't seem to be the case. It's more like a group thing happening, environmental. In the Human Microbiome Project, that's another, of course, very influential evolvement. Unfortunately, the oral cavity has not gotten a lot of attention. There are oral samples, however, they're not they're not dental samples, supragingival samples of the oral cavity. And I will say that the oral samples are very diverse. If you get a salivary sample, you get sampling of what? Tonsils, the oropharynx, the tongue, the buccal mucosa, maybe whatever is shedding from the teeth. But if you think about it, what you really care from a dental biofilm is not what sheds in the oral cavity, it's probably what's adhering or is closer to the tooth, or maybe that other half that's probably closer. Not necessarily, but anyhow, what you get in the oral cavity, the salivary microbiome, is a small subset of the dental microbiome. So we have to do these specific dental studies to get further. So what have we learned? This is not from our work. This is Gary Boris's lab at MIT. And this is one of these new cool staining images that they have produced to show how really the oral biofilm looks like. What is the biogeography of the oral biofilm? And here, what I particularly like is the structure. The purple species is a new species, a relatively newly described species that is called Corynebacterium matruchoti. And this species was not on the radar for many years because it doesn't have really pathogenic properties, but it does create the foundation of these corn cob formations of the dental plaque. And the bad guys, not all the bad guys, but the streptococci, and we know the mutants are bad ones, live on the surface of these formations. So for many years, we have been focused on trying to eradicate the strep mutants on the side, on these sides, and we didn't know where they were. Whereas now, if you think of it more from an engineering standpoint, you may need to do something else to pull the rug under their feet by understanding how they're structured. So understanding the biogeography of the microbiome is really cool and very influential, I think, for potential treatments. And I'm going to show you later one of our studies where we are trying to leverage that knowledge of where species like to be and how they interact together in a geographical way, not only just in their near presence. And this again, another example of the Corynebacterium matruchoti and streptococci living at the very end of these formations, which is really neat. When I went to school, that information didn't exist. We thought we had early colonizers and late colonizers. It was like a soup of things or layers, but they're not layers. They're very well organized. I mentioned about our approach to conducting population level studies and deep phenotyping studies. This is our study population in North Carolina. Every dot is a group of participants. So we enrolled 3 to 5-year-old children attending public preschools, Head Start, in 86 out of 100 North Carolina counties. We would like to go to every county, but we didn't have the funding. We had already exceeded we stretched more than what we could do with funds versus enrollment. So it's not a representative part of the state, it's just a wide coverage of it. We reported a couple of years ago the study, the cohort profile, so you can read more about it and all the data that we collected. I mentioned we enrolled slightly over 8,000 children, and we did clinical exams and biospecimen collection in 6,500 of those. And the mismatch is because you may go to their school, but they're not there. It's a very mobile population. They're sick that day, or they have moved. The core data of our study was the clinical measures. So we did extremely detailed dental characterization, and this is because we're in pediatric dentistry, so we did everything that you can do and more from a clinical standpoint. measuring not only cavities at the individual surface level at different levels, but also developmental defects of the enamel. Sometimes teeth come in that way with imperfections. And these imperfections are very important to know because they predispose you to get a cavity, and also they create these anomalies on the surface that arguably affect what microbiome will, or the biofilm that will grow on them, because they're not as smooth as others. Questionnaires. We collected salivary samples for DNA, and we genotyped all of them. We had the funding to do the genetics on all of them, so that has been done. And we have publicly made the data publicly available in dbGaP, 6,500 children. And we collected microbiome samples from all of them, but we're not funded to do all the work. So we only did 5% of the cohort, which is 300 children. We did whole genome sequencing shotgun, RNA sequencing, and metabolomics of the plaque. So we can study that. So these are pilot for a next wave of the study where we're hopefully going to go back and do the microbiome in the remaining 6,200, ambitiously thinking. So it was a big operation. It took us 13 months to do the data collection, and we estimated about 180,000 miles traveled among the entire team to get to all the sites. We had to troubleshoot a lot. We even collected water samples from their houses, so we could measure how much fluoride is in there. And along the way, we developed a new machine learning geographic information system informed imputation method to infer neighbors' water fluoride content based on their other neighbors. It was really, really cool because we didn't get complete data from the fluoride measurement. So we had missing data points, but we're able to capture a lot back of that. Alongside our kind of sharing, as I mentioned at the beginning, we made these protocols publicly available. And these detail the microbiome part, the clinical part, and the genomics part. So these are made available as a cookbook, basically. We even say what you should order, what supplies you should order to get them, and what software was used, what parameters, all these things. So these are freely publicly available. On the microbiome side, we had to do some extra things because we frequently were far away from any lab or any clinic. And we had to obtain and freeze on the spot, especially if you're doing RNA-seq and you want to observe, to maintain high quality nucleic acids until you get to a lab or a storage facility. So we had these cool boxes and portable freezers that go to -20 to collect samples in sterile toothpicks and freeze them on the spot until we get back to a lab to store them in -80. So we had to do a lot of this work to verify that this actually worked all the way, because otherwise the NIH would not believe us that it's worth investing in this, right? So it was a good thing because they funded us to do all the microbiome work and show that, look, this approach works all the way until the species identification after sequencing, right? You don't only need to show that, look, I can extract high quality RNA, DNA, and this amount. Does this really work all the way? And we were fortunate to be able to show that. So there was a lot of pre-work, even detailing how much plaque we could see and all these things, like very early work. So this is what we did. I mentioned. So we have these 3 layers of omics for this part: the DNA, RNA, and metabolomics. One of the first things we did was try to understand who is there taxonomically in these samples. And of course, you get these really high dimensional reads. And we are very thankful to Jeff Roach that is supporting the microbiome core and helped us really develop custom pipelines and custom reference databases for the oral microbiome. That made a huge difference because what exists for the gut microbiome, the human and all the, the Chris Hattenhauer work from Harvard works amazingly for the gut microbiome, but for the oral microbiome, we're not getting good results, especially when you get down to the strain level or the gene level if you're doing like metagenomics work. So all of this was based on custom databases and optimized pipelines that Jeff worked for years, really. So we ended up creating these criteria. We said, look, for species to be called part of the core, and core is probably a very arbitrary way of saying things that we can study with some confidence. Okay? And of course, you can put the box lower or anywhere else, but it's something that we defined practically. And we said, look, you have to be prevalent at the 0.01% level, but prevalent in at least 10% of samples. So if you're missing 90%, we're not going to study you. And you have to be abundant relatively at 0.01%. So kind of inclusive criteria. And for metabolites, we used a 50% presence in all samples criteria. Again, there's some balanced arbitrary criteria. And then in terms of abundance, unsurprisingly, the top most abundant in both DNA and RNA was Corynebacterium atoxotic, the species that I showed you earlier, that structural thing, which is not associated with health or disease, but it's very important. And again, this is the one. And then we're starting to do more work with this data. One interesting point was that we had to also defend that doing RNA-seq work for the microbiome is worth investing in. Because you may say, well, okay, I will just study who is there, taxonomy. Why do you need to do RNA? And we found that really the RNA does contribute to more functional awareness of what is happening. And that was evident, especially when we did the correlation between RNA and metabolites. So we found that the correlation, this is what this highlighted box shows, is that the correlation between, as a whole, the transcriptome versus the metagenome is higher when you compare it with the metabolite. So if you want to get closer to functions, you should be accounting for the RNA. And there are some other applications I'm going to talk about later. I will not go into detail in this, but I will say that we also studied what clinical traits are most informative for microbiome work. Again, I will not go into this detail, but I will say that number 2, disease definitions that relied on more sensitive criteria, which means that they capture early stages of disease, and they're more close to where we collected plaque samples, which means it's actually talking about the sites that we studied and we collected samples from, you get a stronger signal among those. So, of course, it makes common sense that the sites that we collected plaque from are more representative for the teeth that they were collected from. So, but again, this shows in the data, and we hope that that might not be the case, because if really you can get the sample from one tooth, you can talk about the whole mouth, would be great, because you can talk about the efficient. But this is not the case. There is some site specificity, which again we think it's logical. Finally, we found that there was human content in all 3 layers: the DNA, the RNA, and the metabolites. Frequently and typically, the human content is contamination, so you want to throw it away because you're studying the microbiome. But Jeff had some ideas, and I know other colleagues had some other ideas, that this is actually useful information, because particularly the RNA that you're collecting from where you collected the plaque sample is almost a host response to the microbiome that is there. So you may do some clever things and study together. This is very difficult because these are sparse data, but it's intriguing. So you set it aside, and maybe you'll do something with it later. Similar with the metabolites, we study the metabolome of what is in the plaque, but we're expecting to find bacterial metabolites, and instead you're finding food items, environmental things, human content, and other things. So so just the metabolome of the dental plaque is itself a microcosm of bacteria, viruses, human, environment, toxins, all of the above. So extremely interesting, way more complex than we thought. Well, initially we thought, oh, we're going to get the metabolome, and then we're going to be bacterial metabolites, we're going to put them together, it's going to work fine. There's a lot of filtering and annotation. Speaking about metabolites, when we did all the analysis just for metabolites and cavities, we found 2 noteworthy things. 2 metabolites were, well, there were more metabolites that were upregulated or downregulated, but fucose was an interesting one. It's the sugar that has some important roles in the formation of the extracellular matrix. And that was associated with higher prevalence of cavities. And catechin, it's a flavonoid, was associated with lower prevalence of cavities. And catechin is found in tea, so polyphenol, I think, tea, some fruit, wine, chocolate. So, interesting. As part of all the exploration of microbiome analysis projects, Wu and her team have spent about 3 years trying to identify the optimal methods for metatranscriptomics analysis, differential analysis approach. So what they did essentially was basically saying, look, how do you do RNA-seq analysis? Well, there are methods for single cell sequencing. There are methods for human RNA-seq. You have single organisms, it's fine. But when you have multiple organisms, multiple bacteria, many zeros, and you don't know even what's the exact proportion of what organism in the community, many things fail when you're trying to do the analysis. So they essentially did a comprehensive analysis of all possible methods to do RNA-seq analysis in microbial communities. So they benchmarked all possible ways and all possible methods here on the left. And they did that using our data, the Zoe data, using the Lloyd Price data from IBD, it's Nature 2019, I think, and simulated data. And this is a preprint, which you can go and read, and there are some specific recommendations for what methods to use, depending on the effect sizes, the proportion of zeros, and the sample size of your study. So no model is perfect, but there are some recommendations. Further, there was one application where we tried to use microbiome to predict or associate it with metabolites. And that has been published. It's just an interesting application where you can see again how using oral data, our data Zoe 2, and gut microbiome data from Lloyd Price, how that works. Just a quick and say nice application. Another quick application that I will talk about is the slightly integrative approach. And this is essentially modeling the ratio of RNA to DNA. And several years ago, when we talked about this with Dee, we said, okay, how do we integrate this information? And we said, well, if you have RNA and DNA, the null hypothesis is that a species in a given community will be as abundant in DNA as it would be abundant in RNA, or about equally ranked. Why would they be different under null hypothesis? If a species is very active, will be higher ranked in the RNA data than the DNA, and vice versa. If somebody is highly in DNA and nowhere to be found in RNA, they're either dead or lazy or unimportant, however you want to see it. So let's then model the DNA to RNA to DNA ratio. So we had to develop, and they had developed a model to do that, because that model in this data is basically didn't exist. So actually we got a grant, an R03 grant, to develop the model. And as one example, I will show you here that in the context of dental caries, we found one of the most differentially RNA-DNA ratio species was this Streptococcus oligofermentans, which is a caries protective species because of its properties. So because actually uses lactic acid and has this very, very beneficial role. So it actually validated existing knowledge that you can use this ratio to identify functionally important species in the community. An even more integrative approach is what Bridget Lin, who is doing her PhD with me, is including microbiome and metabolome. And for that, again, we use the approach that Lloyd Price used for the gut microbiome. And here, what we did, we essentially created networks, correlation networks between species and metabolites. And you're trying to see, once you've established these networks, how do they change between health and disease? Because you have certain relationship established in health, and these get changed, disturbed, or recreated in a different way in the context of disease. So we're using these networks to study these changes. And you can see in these networks, you have different types of features. You have nucleotides, carbohydrates, bacterial species, in RNA, DNA, all of the above. So it gets very complex. but they're developing again a new statistical model to account for this. I will finish with the latest study that we have, and this was a combination of both taxonomic and functional work. This is the beginning, essentially, our sampling of all the children, sequencing, alignment, all the statistics, and then we did that discovery analysis, as I said, in the 300 child participants, but we sought to replicate any finding in an additional sample of children we had already sequenced as part of our previous pilot. So we said, look, if we find any new associations with species, we want them not only to be true in the first sample, we want them to be true in another sample to add confidence. Because if you're testing hundreds or thousands of things, you will certainly find some false positives. So we raised a lot the statistical confidence threshold by requiring replication and also associations both in DNA and RNA. So again, very, very strict rules. And then once we found important species, we said, look, now we're going to study those in the lab. We'll do all these functional virulence assays. We will study their biofilm, we'll image the biofilm, and then at the end, if there's something really promising, we will carry it forward to an animal model. And for this, we used help from many collaborators here at UNC and at UPenn. I will not go into too much detail, but I will say that we did find strep mutants re-emerged as important, but also Selenomonas putigina. It was a new species. And these emerged as the most significant, both in localized disease and person level disease. And this is metagenomics here, so DNA and RNA. So these species are important all the time. And actually, we highlighted 4 of them: Prevotella salivae, S. mutans, putigina, and Leptotrichia white. So we said, let's further study those because they seem to have the strongest and most replicable association with dental caries. And again, these are our collaborators here, but they did tremendous work. And this is one example of the kind of laboratory studies. This is metabolic activity as measured by microcalorimetry. Here we have strep mutants with green, Selenomonas putigina with red. You can see how different metabolic profiles they have, how many hours they take to reach their maximum metabolic activity. Strep mutants is the most active and most quickly active, where others are slow growers, and that happens over time. And this is important to know for planning your studies, understanding how the biofilms grow. The most important finding was that when we start adding them in pairs, this is acid production, so proton rate, individually. So for strep mutants, Selenomonas putigina, Leptotrichia whitei, and Prevotella salivae. Here, strep mutants is the most acid producing one by far, and Selenomonas is not doing anything, basically. But when you add them together, which is this column, acid production more than doubles. So that's very intriguing. So why that happens? Because this one is not doing anything on its own. And then when we actually image them, and this is work from Michel Ku's lab at UPenn, we found that there is something going on with their dual culture. They like each other a lot. They grow in this proximity. And we thought that, well, how does this really translate to clinical when we do the experiment? And it is true that when you do the animal experiment, when you put them together, caries lesions also more than double. So there is something with that structure, that relationship that leads to that higher virulence and clinical caries development. So in these all additional biofilm studies, we found that they do form this extracellular matrix that looks like a honeycomb. So that kind of engulfs both species. And interestingly, Selenomonas putigina is usually a motile bacterium. It has a flagella, so it anchors itself and it moves around if it's left alone. But in this context, what it does, and this is again from our own data, this is Selenomonas putigina swimming around on an enamel surface. This is hydroxyapatite. When you put it together with strep mutants, if it's near strep mutants, it's not moving anymore. But what is not near strep mutants, it's still moving. So when they get together, what happens is they get stuck. And the reason is this extracellular matrix. So this is a situation where you must understand who is there, what they're doing, why it's happening, and how that translates to more clinical disease in the animal model. So this was a pretty good story. And this is provisionally accepted in Nature Communications. I think probably you'll see it next month. And this was an interesting finding simply because it comes from our pilot data. So I'm hoping we can get to produce the full study. And this was our overall plan for this study. I will finish with one map. And here I will say that this is, again, our participants in the state of North Carolina. And the dark green are areas of social vulnerability. There are indices that you can get from publicly available data where they will tell you where there's high level of poverty, and they have other measures. So we do know that more disease is concentrated in areas of social adversity. Okay, we could have predicted that, but we're finding with this work. The interesting thing is that there are people who are in areas of high disease, of let's say hotspots of disease, but they have extremely good oral health. And the opposite happens. There are people who live in areas of low disease, and they're doing poorly. So we're now following up these positive outliers. It sounds like a Malcolm Gladwell sequel, but it's a scientific method to some degree. So we're starting to see what is really different from a social, behavioral, microbial, and genetic standpoint for people who are in an adverse social environment, but they're outliers on the positive side. And we think that's an additional, possibly smart way to integrate or leverage the environment, all the things we have, the biology, and also interviews. We have people who are actually talking to these people and saying, okay, tell me, how is oral health going for you? What do you do? And in most cases, we're finding that it's a matter of routine for most families. Interesting, that's a common denominator. It's just part of an established routine. So this is what I had for today. As I mentioned, feel free to send me an email. I'll send you the slides, as we went through some of them quickly, and I'll be happy to take any questions or discussion.
Always amazing work. Oh. Very nice, very nice. Question, I don't know, in Zoom or here?
In Zoom, if you want to just unmute yourself, you're welcome to do that. There are nothing in the chat at the moment.
I was just curious about, you mentioned that you only pick 5% of the whole population to run multi-omics. I was wondering how you picked the 5% and whether it was randomized or whether you tried to select from every area to represent the population.
That's a good question. So it was random in the sense that it was actually the first, I would say random, it was the first 150 participants chronologically with disease and the first 150 participants without disease. And the reason we do that was that, as I mentioned earlier, it was part of a pilot to NIH to show them that what we're doing is working for the whole study. So we had to do it very early in the beginning of the study to show that collecting the samples that way was worthwhile. And the reason was that this was not initially planned in our initial application. Our initial application was only human genotype. But when we got to doing the study, microbiome was already, it was 2013, 14, we said, well, we should do it. So then to do that, we had to do a pilot within the study. So the fastest way was to, in the early stage, to just pick the first 150 and the first 150, do it quickly, and then continue the study that way. So they're not representative of the whole population. It was just, actually, I can probably find out what regions they were. It was when we were in between August and October 2016. Yeah, it's a good question.
You know, something got stuck in my head. The fact that it took 40 years to acknowledge caries as a non-communicable disease, and the suggestion to mothers to not kiss their babies. So, but my question has to do with the fact that, for example, we know that the way the baby is born, for example, will impact gut microbiome. So there is some impact from the mother, right? So what happens with the oral microbiota in the sense that it has to come from somewhere and it's most probably a mother, right? So in a sense, if the mother has a lot of caries, is the baby or the child gonna have a lot of caries?
So that's exactly part of the problem is that epidemiologically it's true. So moms with and dads with cavities will have babies with cavities. The question is that the transmission or the inoculation, or is that the social environment, what they eat at home? If the mom is eating this, the dad is eating this, and I like, Oreos and everybody's eating Oreos, versus it's the biological connection. So my understanding, and this is what I, talk about, is that it's a matter of balance, right? So everybody's going to be colonized with most oral species from their mom, their dad, the caretaker, and the siblings. And I learned about the ABC concept in preschool, which is a chewing gum that has already been chewed. If you're very good friends, you're going to share this. So there is all this horizontal and vertical. So the exposure to strep mutants and other things will happen, I think, anyway. Whether they will establish themselves in an acidic environment and create a dysbiosis, I think it's a behavioral diet environmental. So you may be exposed by your mom, but if the environment is not conducive for strep mutants to overtake everybody else, then you're not gonna get it. If you're eating Greek yogurt, and I'm Greek, if you eat Greek yogurt every day, probably you have different exposure because you're exposed to different things, right? So regardless of the initial exposure. Again, there are some theories that if you get exposed very early in these windows of infectivity that some investigators said, then you really have to walk uphill to reverse that. But I think clinically and epidemiologically, the influence in oral health of the environment being diet, hygiene, is so strong that probably selects for, even if you have very small proportions of bad species, if you feed them properly, they will outgrow everybody else. If you have a lot of bad species, let's say unfavorable, but you don't feed them, and you feed the good ones, I think the good ones will take over because it's a very quickly selecting environment. But we don't have very, very good data to support that. So it's more like a postulation.
I guess we could draw a parallel with the gut bacteria too, right? Because even if you're born, let's say, by C-section, they say, like babies that are born by C-section have more prevalence of I don't know, obesity or whatever, and they say it's correlated with the microbiota. I guess even if you're born by C-section, but you feed the right bacteria, then there's no reason for that to be done. And so, I'm sorry, go ahead. No, go ahead. I'm gonna.
I have to. First is you have. I'm sure with children, maybe you have siblings. Do you see any associations between, like, if there's a pair of siblings, and do you have any cases where you have siblings living in different households, so you can see if it's more of a routine or if it's more of a genetic factor? My second question, you taking in consideration was the child breastfed or formula fed, and how that affected developing caries later in life.
So your first question is very interesting because it made me think of something. We haven't done the work, but we do have monozygotic twins. We have full siblings. We have and we have that verified from genetics, actually, because we have to build a pedigree. So we didn't ask them who was what, who was related, but we created a pedigree as an adjustment for a genetic analysis. We need to know who is related to who. So we have, I think, 34 pairs of twins, and then all the other family structures. So we can study that. Now, when you get to the 300 with microbiome data, I think there are probably 10. These numbers are small, the related ones, but still it can be done. We haven't done it yet, but I think we should. The one with the additional question with whether they live together or not, I hadn't thought at all, but we can do it because we have their addresses and we know who is who. So maybe we'll do it. Yeah, that's a very good idea. We don't have information to ask them about breastfed or formula fed or not. We had only one question about that was, a very like known risk factor for cavities, whether the child went to bed with a bottle containing anything else than water, because we know it's a risk factor. But yeah, we don't have information beyond that. So actually the questionnaire was pretty short, it was 1 page.
And some children even like nurse to sleep.
Yeah. We didn't have that information. Now these children were now 3, 4, and 5. So in some cases, there's not good information if you go really back in time, because there are multiple children now, things such. But yeah, that I wish we had asked more. Actually, we don't even have, I would say, even more important things, such as if they have a major systemic disease, or if they got, we don't know that here. We did ask them about medications, what medications they received in the last period. So we know who got antibiotics, who could drastically influence things, or other things, but we don't even have a good medical history that might be important, right? So we don't have like they have important things. So there are some things that are missing that we would like to know more, but it's a trade-off between doing a very large study with a lot of moving parts versus a small one where you can go in depth, right? So there are many things I would have liked to do more and differently. But thank you for your first question. I think we will do it, and we will get back to you. Yes.
I also have a question. When speaking of the children's teeth, I'm thinking about the baby teeth that will be replaced later when grown up. So once it's replaced, once it's like dropped and the new one grows, do you see any difference between the new one and the old one? Then because you talk about some bacteria is like they can move. So is there any like interaction between the teeth that's just grown up and with the old one?
Yeah.
So or is there any like the saliva or the food that will help the growing?
So one way to think about it is that, going back to the early slides, dental caries is a person level disease. So imagine you have a disease and you have new teeth coming in. They're coming into an environment that is pathologic or health associated or somewhere in between. So it's like you're replacing units, but it's almost like it's raining and you're bringing new players into a raining field. You may be treating the players, but it's still raining. So they're going to be exposed in the same environment. Now, bigger teeth, newer teeth will take more time to develop clinically evident disease, but the environment hasn't changed. That's why even when we do fillings as a dentist, you say, oh, they got a cavity over the filling cavity, maybe they didn't brush it well. It's because we never treated the disease. It's still raining, you're putting white fillings, and then you didn't treat the disease, right? So it's still the same disease happening. So the other thing, and I'll get to you, the only way that you can have an effect of one tooth to another, if you have an infection in a baby tooth, that infection expresses around the root of the baby tooth. And that is exactly above the crown of the permanent tooth. And that change in pH can sometimes affect the integrity of the permanent tooth. So when it comes in with a defect. It's weaker, so it gets disease faster.
Like, for example, following that question, so for the picture that you put that the child needed an extraction, like how is that recovery process with like what is coming afterwards?
So that child is free. These teeth will come in at about the age of 6.5.
Yeah.
So in the meantime, there's nothing there, and you hope that the infection, if there was infection, didn't grossly affect the developing permanent tooth. And the problem is the earlier that happens, the less mature the permanent tooth is. It's like a fetal development. The earlier stages are way more vulnerable because it hasn't really even fully mineralized yet. And you see that even more in case of trauma. So it hits their teeth and get pushed in, they can really impinge the permanent tooth. And then when they come in, you see a yellow spot or a white spot, dark spot, or sometimes shape differences. So we don't know here, but that's why the extraction is indicated, because you're trying to protect also the permanent tooth.
Following up on the same question, you said it's like more of a disease, and if you don't treat the disease, you're gonna still have the same problem.
Yeah.
That prompted me to think about your biofilm study, and you mentioned that there is one strain of Streptococcus that was beneficial and that would feed on lactic acid.
Yes.
Did you ever did a biofilm study where you actually added this addition, this third species, and see how that works, and if it could be like a way of like helping to eradicate biofilms and like giving like a certain like oral probiotic.
Yeah, so actually one of our collaborators, Wayne Ribeiro, has gotten this finding, and she's doing that work. I don't know exactly what work, but she's working with oligofermentants as we speak. And I think she's going to present it in a meeting in somewhere exotic in the summer. So she submitted an abstract. So maybe we'll get back to you on that. But yeah, that's promising. Another thing that is happening, and I can say that very generally because I'm involved in some processes related to that, there is a toothpaste being developed that includes a probiotic. It's a biological agent. So it's an amino acid that is fermented by several species of the oral microbial community and helps, it goes into the urea pathway basically. And that leads to the pH of the plaque stay high. So that toothpaste works not with fluoride, which repairs damages in teeth, and some people don't like. It basically enhances the pH, the nitrate and urea pathway, production pathway of the biofilm, so pH stays high and doesn't cause the mineralization. So it's another way to treat. Yeah. Yeah. Well, it's will come to me. Arginine.
So it's an amino acid arginine as a little bit as a prebiotic.
Yeah, it's a food that is used by.
That's really cool because I was wondering like how would you deliver a probiotic and for it to stay long enough in the oral cavity, because we just like take a capsule and it would dissolve in your gut. So how would that work for oral like microbiome?
Yeah, and you know the oral cavity. I think now that's again I'm not using here data. I think that the oral biofilm is a pretty resilient microbiome because it's it has to withstand what? Masticatory forces, temperature changes, acid, all the things that happen, and it's still there and still doing its thing for millions of years, right? So don't want to downplay the strength of the gut microbiome, but it's a way more controlled. I mean, it's an unfriendly but more stable environment in some way. The dental one, you have things happen like catastrophic events like every hour, and they're still there, with their structure, and it's very hard for us to eradicate anything, right?
I work on prevotella, and some of the samples that I have are oral samples and gut samples. And we have this discussing that the oral samples are easier to deal with because I think that because they get some oxygen exposure, they're not as sensitive as gut isolate. So that's what I'm like thinking, like part of their resilience.
Exactly. Yeah, Prevotella is a bad gum bacterium, Prevotella intermedia, and gingivalis, right?
Yeah, we're having that discussion about which prevotellas are good, which prevotellas are bad. Are they good?
Yes.
Yeah.
Awesome. Great work.
All right. Well, thank you all for coming. I would like to just leave you with a reminder that chocolate is good because it prevents cavities. You said that.
I did.
So thank you so much.
Good.